guys. This is a topic that I've been meaning to make a video on for the longest time. Many students have asked for it, many of you have asked for it as well. And this is to go through just some common techniques that are more molecular and cellular biology related that are important for understanding primary literature article involving scientific research in the biological sciences. It could be a clinical research, it could be a basic science research. A lot of our work is going to be focused on molecular and cellular biology techniques, so it's very important that we understand what those techniques are and what are some common ways the data is presented in the shape of graphs or figures in primary literature articles, and finally, how do we interpret these results. So that's the goal of this particular presentation, is to just go through some of these techniques, the most common ones. This is by no means the most comprehensive way, PowerPoint on all the techniques, but it is about the most common techniques to help you understand literature that you read in order to study your specific questions in biological sciences. So let's get started. So we're going to start off by just looking at what are the different levels of biological information that we might be studying as a researcher or as a scientist. We all know about the central dogma, where you start off with the DNA as our blueprint. You convert that, parts of that you need into an RNA copy. And then from that RNA copy, many times you are either creating proteins that are doing work in our body or in our individual cells, or we are using the RNA itself to do some work. So that's the general part of our molecular pathways, the way information flows and the way we are doing all the work. Now, DNA can be copied itself as well. It replicates itself when it needs to transfer that information to the daughter cells as the process of growth and replication occurs, both at individual cellular level and at the species level. You can also have DNA interacting with proteins directly in order to regulate protein function or protein expression patterns and things like that. We can also look at these in the lab as well. In addition, there are certain systems that are applicable on specific organisms. For example, RNA can make more RNA copies of itself in lower life forms and in viruses. You can also have RNA reverse transcribed into the DNA equivalent. Viruses do that a lot as part of their life cycle as well. But the general, DNA to DNA and then DNA to RNA to protein is going to be present in all life forms, regardless of which organism you look at, whether it's a lower life form or a more complex multicellular cellular organism. So that's right up here at the larger picture. Now, this alone is going to not give us the full picture because once this basic step is done, the cell is now doing work, and it's going to do different types of work depending on its state of differentiation, depending on its environmental factors, depending on what we are consuming, all kinds of things. And so there are going to be certain metabolic pathways that are only present under certain conditions, are active under certain conditions, and there will be metabolites that will be created as a response to all the environmental factors and all the internal factors that a cell is exposed to. This directly relates to which pathways are active inside our cells, individual cells, and how they are controlling the various networks inside that particular organ system, and then as a whole on the organisms as well. So all these things are at the cellular level. Once you look at how these different cells are interacting with each other, we then start looking at individual organ systems inside a multicellular organism, the individual as a whole, which will be affected by the sum of all the different cell types and all the different metabolisms that are happening, all the different pathways and networks that are active at any given time, and then how the group of individuals would affect populations and ecologies. So people, researchers, people in science, researchers of various types are going to be studying one or more of these systems at any given time, depending on their interest, depending on the question they're trying to answer. And all these parts, all these steps are important things to study about a question. Now, we are going to be, because we're going to be focusing on cellular and molecular side of things, we are going to be staying within that framework. So we're going to be looking at the genome, which is at the DNA level, the transcriptome, so DNA, the genome is going to be the same for every single human, for example, right? Or every single, the general sequence remains the same, the amount of DNA remains the same. A lot of different, the number of genes that are present remain the same. However, they will be in different flavors, right? They'll be mutated, or there'll be different variants of each gene that are present in individuals. So there's going to be variety over there to observe and to examine. This is something that geneticists do all the time, right? They study the DNA and its expression patterns, the different alleles, different flavors that are present, blue versus brown eyes, or any other sequence that they're looking at. Then we move next to transcriptome, which is the expression of that genome. Which parts exactly are expressed in a given cell type, in a given organ, in a given system, and how much of that transcript is present as well. So it's going to look at not just which ones are there, but how much of that particular transcript is there. So that is what we call transcriptome, examining the transcriptome. And that's essentially because the DNA to RNA is your transcription process that is occurring inside the cell. Next, we get to the proteome, which is the study of proteins. And you can now study the proteins at the level like we did with the transcriptome, just generally speaking, which ones are there and how much of them are there, and whether that number changes when you have different variants, whether that number changes when you have normal versus mutated, whether that number changes when you have differentiated versus undifferentiated. Over time, all kinds of different systems can be looked at, all kinds of different variables can be looked at while looking at these proteomes. We can also look at how a sequence at the genome level changes the, if you have a change at the genome level, how it changes the level or the type of transcription that's happening, whether that gene is transcribed or not anymore, and then once transcribed, whether or not it makes the same protein product, or if that protein product is now altered either in size or shape or activity as a result of that change that happened at the genome level. So they are all kind of interconnected, right? One step further from that, within the proteome, is examining modified proteins. Now, proteins, once they are built, it's not always the final active form. Many times you actually have to activate them through further modifications. These could come in the shape of phosphorylations of particular residues on your proteins. It could come in the shape of glycosylations or addition of sugars on your proteins. It could come in the shape of acetylation, methylation. A lot of different things can happen to your proteins, just like different things can happen to DNA in order to activate them or inactivate them. So we could look at these, what we call post-translational modifications, and see if they are altered by the variable that we are examining, by our independent variable. And then finally, we can look at the overall effect of whatever environment or whatever stimulus we are using as a variable, and see how that alters biological function by changing the metabolic pathways, by changing the different networks that are affected in that particular cell, that are active in that cell. And that study, looking at the activity or the function in response to an environmental stimuli or in response to a independent variable, is called metabolome. So you have genome, transcriptome, proteome, and metabolome. So if you were to examine at the genetic level, let's say you have about 25,000 genes, you have, in using those 25,000 genes, you have about 1 million different proteins that are present within our cells at any given time, or in our body in any given time. And those proteins are going to affect different metabolic pathways. And there are about 1,000 different metabolic pathways that are known and that are, that we have already started to look at, and that one of these proteins would be part of. So that's something to keep in mind as we move forward. So let's start by looking at, well, what are the different levels at which we can utilize cell and molecular biology techniques. And then within each category, we will talk about the different techniques that are appropriate to use at that level. So we're going to start off by just simply saying, okay, so let's just look at some morphological features within our system. What do we mean by that? Well, when we are looking at a morphological picture or morphological features, all we are looking at are what we can observe under the microscope, and we can look at changes in shape, we can look at changes in size, we can look at whether something has changed in the way that cell is observed by us, whether it has more bulk in density, whether some part of it is breaking down, whether there is more cytoskeletal structure, more protrusions, anything like that would be part of morphological analysis. Whether it has multiple nuclei, single nuclei, are they misshapen, any of that issue. So you can do that obviously by actually making physical observations yourself by looking under the microscope on either cells or tissue sections, on slides, or an organism in real life if it is a large enough organism, and you can make these observations. You can make these observations looking at the entire tissues or the entire specimen that you have in hand. You can do that just by looking at the central region or the core of the region. You can look at just the edges around the membrane, and that would be your rim analysis, or you can look at both, right, and kind of combine that info together to get the full picture. We could definitely do a lot of that by ourselves, by making actual physical observations ourselves with the help of microscope, but we can also use machine learning on computers to do that work as well. And when we do that, we can actually make a lot more observations. There are things that we cannot do with naked eye that we are able to do once we use computers for those image analyses. In that case, you take images of each one of your specimen, and then you train your computer software to examine, to look for, to find key features that you care about, to classify them and characterize the cells according to them. It could be whether they have protrusions or not, whether they are concave or circular, what's their aspect ratio, is there any variability in their size or area or density, Are there any features that are different in the sense of like skeletal organization, like I said, protrusions, or if they're able to now be mobile or move? All kinds of different things can be observed using a system like that. So let's see what some of these observations may look like. So here is an example of two different ways that we can get these images and then make our observations. One of them is what we call bright field. In bright field microscopy, you just have images of cells. These could be stained or they can be just unstained cells, and what we are going to be looking at are the cell bodies and the nuclei and the nucleoli and whatever else we can see with the help of our typical compound microscopes. These images are shown at 400x, but you can look at them typically all the way up to 1000x or sometimes even more with the help of today's compound microscopes. When we look at those, we can look at obviously the changes in shapes of the cells, right? We can see if there is single or multiple nuclei within the cell. We can look at whether there is cell division happening in any given cell or death happening in any given cell. So there are lots of morphological features that can be looked at simply with even just unstained images. We can also examine if the cells appear to be dying in a particular way, for example, right? If they have protrusions, if they're differentiating, changing over time, any of those things can be done. Now, one step above that would be using either stained images or better yet using fluorescent images. When we use fluorescent images, here are some examples of using that. DAPI is a stain which stains the DNA inside the nuclei, so you can clearly see the nuclei and you can see, for example, if there was a cell dividing or dying, you could clearly see what was happening in it. And here, when it's like super intense, you can tell that's your DNA. It's getting it could be getting fragmented or it could be dividing within the cell. So you'll be able to watch that and monitor that. In this assay, it's called the COMET assay. Again, it's staining the nuclei, so it's staining the DNA content. If the DNA is nice and intact and there is no problem with it, and just like, there are your healthy cells, so all the cells, since they're healthy, you see normal DNA content, you see nice, clean nuclei, nothing happening that is too messed up. When you start looking at cells that are kind of getting disrupted, that are stressed, that are beginning to die, you start seeing fragmentation in the form of this kind of comet-like structure forming, and that indicates that there is fragmentation within those cells. Within the cell in the nuclei, the DNA is getting fragmented. You can also look at different cell body structures. You could stain for cytoskeleton, you can stain for the membrane, you can stain for ER, mitochondrial kinds of things with different fluorescent markers linked with antibody for a protein that is present only in that organ system, for example. Here we've used the rhodamine dye, and that gives you this green fluorescent color to examine presence of particular protein that is definitely disappearing as we stress the cells and make them undergo some type of cell death, most likely apoptosis. So using a mixture of bright field and fluorescence, and using markers with different fluorescent colors, we can examine lots of different features at the same time and see them in motion, see how they are different within the different treatments. Now, obviously, like I said, we can analyze this type of data using image analysis, digital image analysis, using the help of computer programs. So here is an example of some stained cells in bright field, but these are stained with a blue dye. and we trained the machine so it was able to identify all the cells that were in the system. That's why these are marked in black, right? And you can see the cell bodies. It should pretty much mimic what it saw over here. When it is done properly, you should be able to get every single cell individually identified. And then over here you see just the boundaries of these cells without the fill. And if you were to go up close, we would see a number inside it to show the number of cells that it was able to count and find. The beauty of this is that we as a human being would not have been able to count and simultaneously look at the cell area, looking at percent cells with like different types of shape or roundness or size or volume. It would have, and if we were to do it for every single cell on the slide, it would have taken a tremendous amount of time. But using the computer softwares, if it is trained correctly, it will do that for you within seconds or minutes, depending on the complexity of the analysis that you're running. And what it will give you will be, you know, just in this image, for example, I was able to get how many total cell count cells were there on that image, what was the cell area for every single one of the cells that were there, what was the biggest and the smallest dimension of every single cell that was on that slide. I was also, because I had set up a separate program parameter to figure out the nuclear size, to identify and figure out the nuclear size, it was also able to give me the nuclear size as well. So that's a tremendous amount of information from a single slide with a very large N number. So here is an example data from one of my papers looking at morphological examinations in this particular set of images. Here we have some liver sections from mice. You have some young mice, some old mice, and some old mice that have been given a GABA supplement to help to improve liver regeneration rate. And we have pre and post partial hepatectomy samples. So before and after some of the liver was resected, or 2/3 of the liver was resected in these mice, we have these images. Now, looking at these, yes, I do see some differences here. Yes, I do see some cells dividing, but I don't see like a very big difference that I can catch right away. It does appear that maybe these nuclei are a little bit bigger in size in the GABA-treated old mice, but nothing really jumps out outside of that observation. And post-hepatectomy, I do see similar kind of larger nuclei as well. So that's something that I can kind of sort of see in here. Not sure if it's statistically significant, but using image analysis, we were able to see that yes, indeed, 2 days post-hepatectomy, we are able to see an increase in nuclear size in young and the old mice, regardless of the fact that they were older. Yes, it was a little bit less dramatic in the old control mice, but you still saw that those cells were trying to compensate for the fact that 2/3 of the liver was gone. In old GABA-treated mice, they already were larger at the beginning of the experiment. So because they were already treated with GABA, they were already kind of primed for that regenerative potential. And they actually reduced in size post-treatment or post-resection, 2 days post-resection, indicating that maybe they had already gone through cell division and now they were smaller. Maybe that is possibility, especially as we see some cells that are either just finishing up cell division or are actually doing cell division as we look at them. We can also count the number of nuclei that I have per millimeter square in that tissue section. And in the young and the old mice, control mice, we see that there were less, right, nuclei 2 days post partial hepatectomy, but those nuclei were larger in size. On the other hand, in the GABA, there's a dramatic increase in the number of nuclei 2 days post resection, while the size decreased somewhat to the typical level. So that provides me insight into how GABA may be affecting the regenerative capability of these liver cells when we treat it before stressing the liver. Here's another example. These are also tissue sections, and in these tissue sections we are able to identify different types of cells that are present within the tissue. This is normal brain. We can definitely see a difference between that and something aggressive brain tumors like astrocytoma or glioblastoma. I can see blood vessels, normal blood vessels. I can see some infiltrating cells that indicates that something is happening beyond the typical brain. I can see proliferation and expansion of these areas of the cells. There are more cells here than there are in the normal brain. And the blood vessels are becoming thicker and bigger and more disorganized. So there is lots of information again that I can get simply from stained slides. What you're seeing here is an H&E, hematoxylin and eosin stained slides. And they allow us to see the nuclei and the cytoplasmic matter underneath in good contrast. Next, we're going to continue looking at these cells at a morphological level. And again, here we have cells that have been grown in culture, in vitro, which is outside the normal environment, outside the native environment of those cells. So this is in a lab setting. And you can again see just visually changes in their size, shape, behavior. You can see decrease in cell number, increase in cell number over time. So you can see that stuff. We can quantify it, right? We can quantify this information as well. So here you have cells, they plated a given number of cells, and they looked at their viability over time, over 8 days. So they started with a given number of cells and they said, okay, let's see how they responded on day 8. If I saw more cells, less cells, when they were treated with a particular agent, and you saw a reduction in viability. So we can run cell viability assays. The simplest cell viability is simply that you can plate different number of cells and see the growth curve. You see them growing and then eventually plateauing off. You see a growth rate. You can figure out what's the doubling time of a cell line is, for example. Here's another example. They had different types of cell, number of starting cells that were plated, and they looked at how quickly they grew, how well they grew over time, and they were able to see that the 200 cells, when they were just seeding 200 cells, that was low enough cell number, that over 8 days those cells continue to grow in a linear manner, still staying within the log growth phase, while when you had more cells, sometimes that slowed it down because there were already too many cells in the environment. Here you have another way that we can look at cytotoxicity of a drug, for example. Here the cells have been treated with a different concentration of a compound that is cytotoxic to these cells, and we look at their viability 48 hours post-treatment. Typically you do that by first staining the cells. You can visibly see less and less cells in each culture, and then you can solubilize the dye. This is could be a crystal violet dye, for example, and the dye would simply stain whatever's on the plate if it was a crystal violet dye, and then you would solubilize the dye back and you would see how much of that dye was present in each one of the conditions. You could also do the opposite where, or you could also look at MTT-based cytotoxicity assays, where now you're looking at metabolic activity of the cells, and you see the viability of the cells in response to a particular drug, and you are able to see that, I think this is supposed to be, percent death, that you are able to see that the drug either protects the cells or kills the cells over time. As the concentration is increased, the cells do not fare as well. You have less and less number of cells and they're less and less metabolically active. So here you can see that, just the DMSO itself didn't cause too much death, but with these different compounds, some compounds caused a lot of death, some compounds caused little death. Here is another way that you can look at cell viability again. So like I said, this is crystal violet stain, and in the crystal violet stain you have your control cells, you have treated cells. Just visibly, I can see there's a lot less cells here than there are in the control. The control grew very beautifully. They were nice and healthy cells. Here the cells look stressed and dying, and very few cells are remaining. When we quantify it, we will quantify it using a graph either like this, where we look at percent viability, or we can look at percent death, depending on what it is we are looking at. Here in B, they're using another assay, which is called a colony assay. You seed the cells at super low concentration, so they have plenty of place to grow. And then as they divide, they start to look like little tiny colonies. And you count the number of colonies that are present in your sample. This could be just cells growing on a tissue culture treated plate with nothing different. Or you could be looking at migration of cells, in which case we could be doing an assay like Boyden chamber assay, where you have a layer of agar over there, so the cells are kind of suspended within the agar, and they have to make kind of these colonies, a little ball of cells, a spheroid of sort. And then what you do is you count the number of colonies that you get with and without the compound that you're working with. If you have cancer cells, for example, you expect them to make lots of colonies. And if you are giving them an anti-cancer drugs, you should see significantly less colonies post-treatment. And so you can make a bar graph, for example, looking at the average number of colonies between control and treated cells. And you could look at statistics around that. You could also look at absolute cell counts in a system like this, and you can use, again, your control compared to your treated cells, and you can examine how the drug changed the number of cells that are present there. Here are some more examples of looking at cytotoxicity cells with the change in color of reagent or the number of colonies. So here you have cell viability, you have your control, lots of healthy cells touching each other, growing very well. Once you start adding the drug, it is not as healthy. They're all looking a little bit stressed and look like they are dying. And they are going to reduce in their cell viability percentage over time. So the reason you are looking at numbers above 100% here is because you're starting with this number of cells and you're looking at whether it is going up or whether the cells stay the same or whether the cells start to die compared to beginning. Now here you have optical density due to color. So if you take the color off of them, solubilize it, and then look at it, you can see that there is more and more color in this case as you have more and more drug treatment. If this was a metabolic assay like MTT, you would expect that number to go down as you go up to indicate that the cells are dying. Here you have the colony count, and you can see the reduction in number of colonies in a dose-dependent manner. Here again, if you were to look at absolute cell count, you see a reduction is cell number, actual cell number, in a dose-dependent manner as well. So there are other, like I said, in vitro 2D methods that can also be used, not just looking at cell viability. Some of those are just using time-lapse video microscopy, where you take, you have your cells culturing in the incubator system with a microscope or another monitoring device embedded within it. It would take pictures at given amount of time, every 10 seconds, every 10 minutes, every 1 hour, whatever you want to set it to. And then you stitch them all together to look at your cells over time, over days, or whatever you need to see how your cells are changing in that time frame in response to the treatments or variables that you're modulating or changing. You can also run what we call wound healing assays. In a wound healing assay, you first allow the cells to grow completely, covering the surface of the vessel that you are growing the cells in. You then create a scratch to create a clearing within it, and you watch how long it takes for the cells to migrate, replicate, divide, and close up the gap. You can also do another type of system. There are two different ones that you can do. You can do a cell barrier assay. It's a little bit like your wound healing assay, where you have cells growing and you have a barrier or a break in between, and you examine how long it takes for the cells to breach the barrier and close up the gap. In a chemotaxis done chamber, you have the cells that are growing on one side of your culture plate on a membrane of sorts. Underneath, you're going to have an attractant gradient from one side to the other. You have a area of clearing, and you see if the cells will move from the area of low concentration of attractant to the area of high concentration of attractant. And it will look at the migration or chemotaxis ability of your cells. In a Boyden chamber or a trans-well system, it works similarly in the sense that you have a membrane that has cells on it. You have the cells grown on the top of the chamber along with some media with the environmental factor, whether it's a drug, whether it's a protein, whether it's something else. The exposure that you are using as your independent variable on the top side. And then in the bottom area, you will have a chemoattractant with the media, and then you allow the cells to incubate and respond to that chemoattractant. As it does that, the cells will move to the opposite side underneath the membrane. You then remove the cells and remaining cells on the top, and you stain and count the number of cells that made it to the across the barrier to the other side towards the chemoattractant, and you quantify it to examine how well these cells are able to migrate without any other help. There are other things that we can look at to examine migration of cells as well. Chemofluidic, microfluidic chip is one of them, where you have a place for the cells to be entered into the chamber. The cells then have to move through the chamber, through various barriers in these microchannels, in response to an attractant gradient until they reach the final chamber. Cells that are adherent and that do not migrate will remain on the top and will not go anywhere, both in Boyden and the proximal chamber. The cells that are migrating, that have the ability to move, will move through the microchannels or through the membrane to the other side and follow the attractant gradient until they reach the highest level, and at that point they will become non-motile and stop overall and just remain in that area itself. Here again is just showing you one of these, which is the wound healing assay, what type of data you get. So remember, you have made this scratch or an area of clearing. You measure it before and after at the at the start and end of your experimental time frame, and you look at the gap width in micrometers, and you can chart that width just the way it is at the start and at the end and see if it reduced over time. You can also look at the area that the gap is in by manually tracing this space and looking at that area at the start and then comparing it to the end in micrometers square. And finally, you can look at a relative wound density by looking at the ratio of the occupied area in the gap as compared to the total initial area in the gap. And finally, you can count the number of cells that finally migrated and look at that as well. Okay, next, let's look at some 3D cell culture techniques. This is a fast-growing part of cell culture. There are all kinds of ways that we can kind of mimic the natural environment inside our bodies, organisms that we couldn't do before, right? Cells that are just grown individually, just the breast cells, just the hepatocytes, just the endothelial cells are not the way they normally are inside the body, so they will never behave exactly the same way. In order for them to behave the same way, they have to be in combination with the same types of cells so they can cross-communicate with them and create an environment that is mimicking more the natural environment they're in normally. So that there are 3 different ways that you can create 3D cell cultures. One of them is 3D heterospheroids. This is kind of a little bit of a forced aggregation of cells where we are going to be adding some type of potential aggregation materials, aggregation enhancers. We might be providing them with a scaffolding to attach to to kind of promote this kind of function as well. So we select the type of cells that we want to get in the spheroid together, and we mix them up and let them create that, kind of force them to create that initial aggregate, which is a haphazardly used like mixed cell ball essentially, and then you allow them to grow and mature and proliferate in culture. As they do that, the cells are going to rearrange themselves into a more organized structure, and that structure, that mature spheroid, is going to have then different areas where you can see different types of cells interacting with it. You can have immune cells in there, you can have oxygens and nutrients permeating through it. You might have areas where nutrients are not able to get through and maybe hypoxic if you don't provide it with the right type of system to work with. Another way is to create an organoid, and in an organoid you have the culture of cells in some type of an extracellular matrix environment. It could be matrigel. It could be a 3D bio scaffold, right? It could be a bunch of different things. You have the medium. You have that media. You have the cells embedded individually, like they are all kind of spaced in single cell suspension in this matrigel or in this extracellular matrix type environment. You provide them with growth factors, you provide them with differentiation factors, and allow them to mature over time. Once they mature, you look at how they are forming, what type of structure they're forming, who is talking to who, how are they arranged, and you examine that more closely. And what you will notice is that the cells will organize in a particular way based on their typical function. So this was a mixture of cells that contained some fibroblasts, some cancer cells, some immune cells, some matrix proteins. And what you got is what we call a cancer organoid right here. With the cancer organoid, the cancer cells all kind of coming together to create this tumor type ball of cells, and the fibroblasts and the immune system cells on towards the outside interacting with those cancer cells. A third way that we can create these organoid is a cell on cell kind of co-culture situation, where you have in this case we have a microfluidic chip or some other type of platform mimicking that situation. You see the cells in combination with a possible scaffolding, extracellular matrix, hydrogel, something for them to kind of be embedded within. You add the immune cells there to see where they end up, and you allow them to move around within this culture format. You allow for them to move as they need to create that culture. And then there are two different ways that you can have that. If you let them make a co-culture by themselves, you would get very similar to how we got the spheroids, where you'll have the cancer cells towards the inside, the fibroblasts making a ring around them towards the outside. If, however, you kind of design the setting for them, you could have one type of cells on, in this case, you have fibroblasts in one channel. You have the cancer organoids in another channel, but what you will notice is that there will be some crosstalk happening between them as they begin to grow and get signals from each other. Next, we are going to now move a little bit more into instead of cellular techniques, we're going to talk more about the molecular techniques. So the first molecular technique that we are going to talk about is immunohistochemistry. Immunohistochemistry is where we can look at some tissue sections from whatever system we are working with, from an organ system, from the particular model organism or humans that we are working with, and we are going to be staining for a particular one or more proteins of interest using antibodies against those proteins. And in this case, you add a primary, you start off with your tissue section, you go through a step of preparing that tissue to bind, to have the antigens open and available so the antibodies can bind to it. You add your primary antibody, it's going to bind to your protein of interest. You then add a secondary antibody against the primary antibody so that it can enhance that signal. This secondary antibody usually contains some type of a fluorescent or colorimetric marker that you will actually visualize. And then you can see either when you add the substrate for color development, you can see color in the areas where that particular protein is expressed. You can look at it through microscopy, you can take images, you can then use computer systems to do image analysis. Any one of those things can work. Here is an example. The top panels are hematoxylin and eosin stained cells, so they are just looking at the general morphology of the tissue sections we are examining. The bottom section are the neighboring sections of the same tissues, but this time what we are looking at are levels of attractant expressed within those individual tissue sections. And so you have the background, staining is just that H&E, and then we are looking at the brown stain. The brown stain depicts the attractant protein. And you can see that from normal to grade, low grade versus high grade, you get a increasing amount of attractant being expressed in and around the tissue, individual cells within the tissue, as the tumor gets more and more aggressive. And in the schwannomas, which is not a type of astrocytoma, you do not see that increase. So it's not just because there's a tumor, but because there's an astrocytoma that you see attractant there. Attractant seems to be related to that. Now again, just like we talked about before, if we just have tissue sections, of course we can make observations ourselves, but we are looking at just one sample at a time, from one tissue, from one mouse, from one human, tumor, and that's going to be a very laborious task to get a lot of information for a lot of patients. So instead of looking at just one tissue on a slide, nowadays we can create tissue microarrays where you can examine many cells, many tissue sections at the same time on the same slide. And what we do is basically we take cores, small tiny cores from various tissues, and we place them on the slide. We embed them within wax to create a little block with lots and lots of tissues from lots and lots of sources represented. We section them and we stain them at the same time with different antibodies or different things, and then we can compare the expression pattern within them. The backdrop is that you're only taking a small tiny portion of that bigger tissue, so you could be missing some important information. But for certain purposes, this is plenty to get a starting information for. So here's an example. Here you have various tissue, you have a tissue microarray. As you keep taking sections from it, you see the tissue is disappearing over time because you keep sectioning off or taking a piece of it. This is the typical tissue microarray on a slide. This is one where it's been stained for a particular protein EpCAM expression in esophageal cancer, and so this is a tissue microarray representing 118 cases. And what they did was they took 1 millimeter cores to place on top of their slide. And so they had 4 tumor biopsies per case, so 4 cores from their block of tissue, from their block of cancer tissue. And then they also had 1 biopsy of normal squamous epithelium from about 40 individuals to get their control samples. And looking at that, they were able to then make actual conclusions about EpCAM expression and how it affects esophageal squamous cell carcinoma. EpCAM is an epithelial cellular adhesion molecule, so obviously if you have more expression of it, that could be a problem for the cancer cell. And so what they saw was that increased expression was usually seen with increasing grade in most cases. It's a transmembrane glycoprotein that is restricted in its expression pattern typically in adult human tissue. It's only in basolateral cell membranes of glandular or pseudostratified and transitional epithelia. But you do see expression of that reappear in cancer cells. Not all, but some cancer cells. Here is a close-up of what that expression pattern looks like when you take just a tiny 1 millimeter core. You can obviously see plenty of tissue even in that small section and variability within them, and you can grade them based on the level of expression. The higher the expression, the higher the number. And it just shows that the normal squamous epithelium typically did not show any expression of this protein, and then in the cancer cells they were able to see that about 60% of them showed some expression and about 30 of them showed really high expression. Okay, so here is an example of how combining all this type of information can lead to better understanding of a particular problem and better ideas on how to use that information for the betterment of that system. So if we were to combine both, biopsy as well as cell culture techniques to get information, we will be able to not only get the information on that particular patient's tissue itself, but we could also see how those cells are behaving in culture. We can observe them as they divide, as they go through their everyday functions. We can use that information to look for proteins and proteins and expression patterns that are different compared to normal, and then utilize that information to develop targeted therapies or to look for clinical markers as well. So a typical proteomic workflow focuses on using our proteins within the cell, if it is a culture system, or if it is an organism looking at biological fluid, tissues or biopsies, and then using those to find differences in gene expression, differences in protein expression, and looking at how to examine their function from that information. So you can prepare samples from fluids, from biopsies, from cells, from subcellular components. You can separate those proteins once they are extracted through 1D or 2D gel electrophoresis or liquid chromatography. All of these methods are going to separate them by size typically or by pH. And then you can just examine them by staining them either through a fluorescent stain, radioactive stain, Kumasi or silver stain, and look for changes in expression patterns. You could also identify individual proteins using mass spectrometry or cell-D techniques. You can validate those results using more molecular analyses like Western, ELISA, immunohistochemistry, microarrays, and Northern's. And then finally, you can do functional analyses and see what those exact proteins, what type of functions those proteins perform. So we're going to actually stop here and we're going to revisit this in the second part of our lecture. And so make sure that you keep a lookout for that, where I will go through these analyses in more detail. And you can focus on the morphological examination in this first part.